We will learn about the structure and type of RNA. Now, DNA contains the information needed to maintain cell process, provides blueprints will never leave the protected nucleus. But RNA it transmitted data to the body of cell itself, where it may be put to use. RNA plays a essential role in transfer of genetic information during protein synthesis and in control of gene expression. Its diverse functions in living organisms include enzymatic activity, storage of genetic information in viruses and viroids. So RNAs may be genetic, non-genetic, catalytic, non-catalytic, coding and non-coding. Now, thermodynamic stability of RNA structure. Thermodynamic stability tells about the energy, so low energy changes. Primary structure of RNA refers to the sequence of nucleotides. Secondary structure in RNA dominated by the Watson-Crick base pairing. This fundamental interaction between nucleobases leads to formation of double helical structure of varying length. In RNA, double helical tracks are generally short. RNA double helix adopt A form structure which differs significantly from the canonical B form adopted by DNA double helix. RNA secondary structure is generally more stable than its tertiary. Thus, formation of the secondary structure dominates the process of RNA folding. RNA tertiary structure forms through relatively weak interaction between performed secondary structure elements. Now, types of RNA. Within a given cell, RNA molecules are found in multiple copies and in multiple forms. Major classes like RNA, tRNA, snRNA, snORNA, miRNA, xist, scRNA, siRNA, tmRNA, and telomerase RNA. Features of few major forms of RNA present in prokaryotes and eukaryotes are given below. Now, tRNA are transfer RNA, a small, well-characterized RNA with a key role in protein biosynthesis. It also known as adapter RNA. Concept of adapter is to provide the interface between nucleic acid language and protein language, introduced by Crick in 1955. It also participates in non-protein synthetic capacities. They prime reverse transcription in retrovirus life cycle. Now, structural properties. It are the single RNA of 75 to 93 nucleotides present in cytosol and organelle of all living cells. Holley and his co-worker determined the first tRNA sequence in 1965, dictated by their primary sequence. tRNA fold into clover-leaf-like structure, like secondary structure, with well-defined stems and loops that make up the acceptor arm, D arm and loop, anticodon arm and loop, and T arm and loop. Regardless of length of tRNA, the number of conjoined nucleotides remain constant. This is the nucleotide sequence of yeast. tRNA this was introduced in 1965 by Robert Holley and his colleague. It's shown the cloverleaf conformation in which intra-strand base pairing is maximal. The following symbols are like phi pseudouridine, I inosine, and T ribothymidine, D5,6 dihydrouridine. Presence of G double bonded U base pair are signified by blue dot to indicate non-Watson-Crick pairing. There is a site of amino acids, acceptor arms, T5C loop, variable arm, anticodon loop, and anticodon triplet, and the do loop. This is a clover leaf-like structure. The acceptor stem always has 7 base pair and 4 single-stranded nucleotides, include an absolute conjugate CCA sequence. The D stem and loop are of variable length, where anticodon stem has 5 nucleotide and anticodon loop has 7 nucleotide. The variable region has 4 to 5 nucleotides but can contain up to 24 nucleotides. Finally, the T stem always has 5 bases pair and T loop has 7 nucleotides. All tRNAs share the same secondary structure. tRNA is classified according to the length of their variable region, which can be 4 or 5 nucleotide, class 1 tRNA, or 10 to 20 nucleotide, class 2 tRNA. Now, a new class tRNA discovered more recently in many organs serve as the adapter for selenocytes, which is the 21st amino acids. Now, tRNA thought to be fold into L-shaped 3D structure necessary to fulfill their function. All tRNA also share the same L-shaped 3D structure. Now, 3D structure and 2D structure. 3D is like contains all the three sides, length, breadth, height, and 2D contains only two, like length or height, length or breadth. It is. In each cell or organ, there are 20 families of tRNA, one per each amino acid. Isoacceptor tRNA belong to same family and are charged with the same amino acid during protein synthesis. Now, rRNA is the ribosomal RNA component of protein synthesis factory in cell eukaryotes. Ribosome contain 4 different rRNA molecules like 18S, 5.8S, 28S, and 5S RNA. 5S RNA, 16S RNA, and 23S RNA in nuclei. 3 of RNA molecules synthesized in the nucleolus and 1 is in elsewhere. rRNA molecules are extremely abundant. They make up at least 80% of RNA molecule found in typical eukaryotic cell. Synthesis of 3 nucleolar RNA is unusual because they are made on one primary transcript that is chopped up to 3 mature rRNA molecules. These RNA molecule and 5' sRNA combine with the ribosomal protein in nucleolus to form pre-40S and pre-60S ribosomal subunit. These 3 pre-subscripts are exported to the nucleus where they mature and assume their role in protein synthesis. The RNA molecule play in role in first protein synthesis. First 28S RNA's catalytic role. It forms the part of peptidal transferase activity of 60S subunit. Second 18S has recognition role involved in correct positioning of the mRNA and the peptidal tRNA. Now, small nuclear RNA is class of small RNA molecules found within the nucleus of eukaryotic cell involved in the process such as RNA splicing, removal of intron from hnRNA, and maintaining telomerase. They are also always associated with specific protein and complexes referred to as small nuclear ribonucleoprotein or sometimes as SNRPs. Small nuclear RNA is class of small RNA molecule involved in chemical modification of ribosomal RNA and other form of RNA by methylation. Mainly, snRNA is a component in small nucleolar ribonucleoprotein which contain SNRNA and protein. It guides the complex to modification site of target RNA gene by sequence in the snRNA that hybridizes to the target site. The protein then catalyzes modification of the RNA gene. A zRNA is guide RNA function in RNA editing. It found only in the mitochondria of kinetoplast, where RNA are edited by inserting or deleting stretches of uridylates. The zRNA form part of editosome and contain sequence that hybridize to matching sequence in mRNA to guide mRNA modifications. The microRNA form of single-stranded RNA, typically 20 to 25 nucleotides long and thought to regulate expression of other genes. DNA sequence code for miRNA is longer than This DNA sequence include miRNA sequence reverse complement. When this sequence is transcribed into single-stranded RNA molecule, its sequence are reverse complement base pair to form double-stranded RNA hair loop. This form a primary mRNA structure. Pre-miRNA draws a nuclear enzyme cleave bases of hairpin to form pre-miRNA. The pre-miRNA molecule then actively transported out of the nucleus into the cytoplasm by exporting protein. The dicer enzyme then cuts 20 to 25 nucleotides from the bases of the hairpin to release the mature miRNA. The function of miRNA appears to be in gene regulation. For that, miRNA is complementary to a part of one or more messenger RNA, usually at site of in 3' UTR. The annealing of miRNA to the mRNA inhibit protein translation. In some cases, the formation of double-stranded RNA through the binding of miRNA trigger degradation of mRNA transcript through a process similar to RNA interference. Though in other cases, it is believed that miRNA complex block the protein translation machinery or otherwise prevent protein translation without causing the mRNA to be degraded. tmRNA is complex with tRNA and mRNA-like region. It currently found in bacteria, ubiquitous in all bacteria. tmRNA recognize ribosomes that have trouble translating or reading an mRNA and stall. It leaves an unfinished protein that may be terminal in the cell. It acts like a tRNA first, then an mRNA that encodes a peptide tag. Ribose translates this mRNA region of tmRNA and attaches the encoded peptide tag to C terminus of unfinished protein. This attached tag targets the protein for destruction or proteolysis.